Okay, welcome back, everybody. Let's get started for the plenary session. Thank you for sticking through this. It's been a great morning, afternoon, honoring Denise. And I'm excited to introduce Dr. Mick Rugg, who will be presenting a talk on the cognitive neuroscience of aging, looking back and looking forward.
I've got it. Here we go. Okay. I will repeat that joke for the benefit of those at the back. I am acutely aware that I am the only thing that stands between you and getting out of this room. So I am going to talk, I hope, for 40 minutes or so, and then once the anger has died down and Thad has cooled down a bit, we'll be able to carry on with the rest of the the day. So. My talk is entitled Cognitive Neuroscience of Aging, Looking Back and Looking Forward, and that's an extremely arrogant talk which came to me in a fit of inspiration, and I never modified it. First off, it should really be called the Cognitive Neuroscience of Aging and Memory. I have nothing to say today about cognition outside of memory, and it's not even memory, it's episodic memory. So by the time we've finished, things will have got even narrower, but we're starting off on a very broad front here. So, sorry, let's make this work. The first thing, of course, Denise, thank you. We have paid many tributes to Denise already. I just want to add mine, and the best way I could think of doing it was by examples of some of the papers that she has been a contributor to or the leader on over the years, which have influenced our own work and which have popped up in more than one of our papers as citations. So we have said a lot about Denise, but we should not underemphasize her influence on the field and the seminal work that she has done in the cognitive neuroscience of aging and continues to do. Moving on with acknowledgements, these are the people who are either in my lab or were in my lab and who headed up the work I'm going to be talking about today. Josh Cohen, University of Notre Dame, is with us in the audience, I'm glad to say. Marianne is here, Ming Zhu, Sabina, and Tracy, Preston, Song, and Paul have all moved on to better things. But I am grateful to them for heading up the work I'm going to be describing. And I also would like to acknowledge that my current lab, some of whom overlap with that previous slide, but a wonderful bunch of people, fantastic to work with, and we've been having a ball over the last 2 or 3 months, ramping our data collection up to pre-pandemic and actually better than pre-pandemic levels. That's all due to the efforts of these people. I'm very grateful to them. So finally, of course, one has to acknowledge funding agencies. The most important of these is the National Institute on Aging. But we've also had support over the years from NIMH, National Science Foundation, and a local foundation, the Aging Mind Foundation. So what is it that I'm interested in talking about today? Obviously, in a 40-minute talk, one isn't really going to talk about the cognitive neuroscience of aging, or even the cognitive neuroscience of aging and memory. But what I want to focus on as much as anything is exemplified by this slide here. So this just shows individual participant scores on a memory test, which is a pretty pure test of episodic memory, associative recognition. And what we can see is the standard, well-known difference, I won't say decline, because this is a cross-sectional study, but the well-known difference as a function of age in average memory performance, shown here, and very obvious here. And one of the things, of course, that we all want to understand is what is it that is responsible for the fact that older people have worse memories than younger people, And as we know from longitudinal studies, memory within an individual declines at least after the age of about 60. It's somewhat controversial as to when that memory decline starts from different longitudinal studies, but certainly from 60 years on continuing, there is an accelerating trend to a memory decline. What is it that's causing that? And that's an important question. An equally important question, though, is what is it that leads to individual differences even within an age cohort? Why are some people really good and some people really bad at this memory test? And we can see that the variance in this participant population is roughly similar, actually, in each of the different age groups. It doesn't have to be like that. It's like that because we are very selective of our older people. And we try to eliminate from our cohort anyone who might be at high risk of or suffering from pathological cognitive impairment. I don't want to open a can of worms and say, what does that mean? But obvious things like mild cognitive impairment, Alzheimer's risk, and the like. So we'd also like to know about individual differences within age cohorts. And also we'd like to know about the interaction. Are there factors that explain variance in memory performance as a function of age? And those are the things that we've been particularly interested in my lab for a few years now. And we sort of very lazily, or I very lazily, classify these in two different ways. Factors that account for variance in performance independently of age, we're going to call age-invariant variables. So if we find some variable that we can measure in the brain that co-varies with memory performance and does so to the same extent in a 20-year-old as a 70-year-old, that's an age-invariant factor. An age-dependent variable is one which accounts for variance in performance as a function of age. And in our hands, so far, the only age-dependent variables that we've found are ones that will account for variance in memory performance in older people, but don't seem to do so in younger people. We haven't found things the other way round. There's no reason in principle why those factors shouldn't exist, but we just haven't found them. So we make a distinction between age-invariant variables accounting for variance in memory performance, and we see these as being extremely important for an understanding of the general cognitive neuroscience of memory. Age-dependent variables, we think, are likely to give us some traction in understanding about variables that emerge in later life and mediate memory performance. This does not mean that these are variables which are telling us anything about pathology necessarily. And to date, all the work I'm going to talk about is cross-sectional, so we don't even know if these are variables that should correctly be identified with the aging process, as opposed to something like a cohort effect. Don't know why I'm carrying on now. But anyway. All right, so I'm going to be talking exclusively about task-based fMRI today, and I'm going to be talking about fMRI experiments that we've done while people are undertaking memory tests. Most of these memory tests in the encoding phase, people do not know their memories are going to be tested later. Having said that, we can find no difference depending on whether encoding is incidental or intentional. And we think the reason for that is that even in our intentional memory tests, where we tell people their memory is going to be tested, they are always given a challenging study task to do. So they're always engaged in the study task, regardless of whether or not they know their memories are going to be tested. And we can look at encoding processes. We have not yet studied what one might call storage or consolidation. That is entirely possible to do. And we have focused also on retrieval. And importantly, and what's going to be important for the second part of my talk, we also look at encoding retrieval interactions. This three-stage model of memory, which is known and beloved by cognitive neuroscience of aging people everywhere, is a little misleading. We've known for decades that encoding and retrieval interact with one another, and although we can write them out as separate words, in reality, it's not quite like that. I'm going to talk about that in the second part of my talk. So let me give an example of the kind of experimental tests that we use and the kinds of contrasts that we look at in our fMRI data to get some kind of notion about the neural correlates that are predictive of successful memory encoding, where we predict successful memory encoding is operationalized as being later able to remember something as opposed to not being able to remember it. And also the contrast that we look at retrieval. So this is our associative recognition task. People lie in the scanner and they study pairs of words. And their task is to make a judgment about which of the objects denoted by this word would fit into which. Sometime later, they do a memory test and they get 3 kinds of word pairs. Intact pairs, just a repeat from the study phase. Rearranged pairs, which are words that have previously been studied but were studied on different trials. They're now presented together. And new items. And the attractive feature of this memory test is that if we contrast, the only way that you can solve the problem of distinguishing between these 2 kinds of pairs is by remembering the association between them. All of the items in these 2 classes of word pairs, intact and rearranged, are familiar. So to make this discrimination, you've got to think back to the study episode and remember were those items paired together. And then we can do critical contrasts. So to look at encoding at the time of study, we can look at the neural activity that is elicited by test pairs that are subsequently correctly judged intact compared to test pairs that are incorrectly judged rearranged. So if I get an intact test pair and I say it's rearranged, I can be confident people have forgotten the original pairing. So I'm contrasting successful and unsuccessful episodic memory. At the time of retrieval, we contrast activity elicited by intact test pairs that are correctly judged intact relative to intact pairs that are wrongly judged rearranged. So again, successful versus unsuccessful retrieval. And we operationalize retrieval monitoring by the opposite contrast. Those intact pairs that are wrongly judged rearranged relative to pairs that are correctly judged intact. And our argument there is that people are going to be spending longer reflecting on and iterating and trying to come up with a memory decision about items that they're uncertain about whether these are rearranged, intact or not, than items that are clearly recollected. So I'm going to focus on only 1 or 2 effects in this part of the talk. The first is 2 regions of the brain that have been heavily investigated in cognitive neuroscience of aging studies, the inferior frontal gyrus and our old friend, the hippocampus. And this exemplifies the distinction that I have drawn already between age-dependent and age-independent effects. So if we look at this so-called subsequent memory effect, these are regions in the IFG that are more active when people process a word pair they later remember than a word pair they later forget. This is called a subsequent memory effect. And in older people only, the size of that subsequent memory effect is correlated robustly with associative memory performance. That is, if you're a participant in one of these subjects and you're an older participant, the bigger your subsequent memory effect is in the inferior frontal gyrus, the better is your memory performance on the experimental task. By contrast, in the hippocampus, we again see a robust relationship between subsequent memory effects and memory performance, but now this is age invariant. And what I'm plotting here is the residuals having controlled for the effect of age, so we can simply see the pure effect of subsequent memory effect on memory performance. And the reason that we're entitled to predict these residuals, or to plot these residuals, is that the interaction term in the regression model is non-significant. If you want to, I won't rant, but I will say that it is not, in our opinion, appropriate to plot a bivariate relationship across age groups and then think you found a relationship between memory performance and anything else. You found a relationship between age memory performance and something else. And also, it's not appropriate to do a partial correlation across variables that actually interact with the predictor variable. But in this case, that's not the case. So that's what we found there. Here's another experiment that very quickly is a different way of operationalizing episodic memory. So in this experiment, we had people study pictures, and they had to make one of two different sorts of encoding judgments on this picture. Either it was bigger or smaller than a shoebox, or it would be found indoors or outside, or outdoors. And they were cued as to which judgment to make just ahead of seeing the picture. If it's a cow, I presume everyone would agree that the answer is, yes, it's bigger than a shoebox, then yes, you're more likely to find it outdoors. And then at test, we have people do two different memory judgments. They first off tell us whether or not they've seen this picture before, and we have them make a confidence rating for that. And if they say the item is old, they have to tell us whether or not it was judged for its location or judged for its size. And again, they make a confident judgment. And the critical point about this is that in this paradigm, we're able to contrast encoding effects and retrieval effects whilst controlling for overall memory strength. So we only look at those items that people make highly confident judgments to. And this gets some way, not completely, but it gets some way away from the confound that otherwise exists between memory strength and age. All other things equal, older people have weaker memories than younger people. Controlling for that by getting confidence judgments gets us not completely, but a lot of the way away from that problem. So again, we can look at encoding effects. We can now look at encoding that predicts source memory, and we can look at encoding effects that predict item memory. Two different things here. And again, our old friend, the IFG. If we look at the subsequent memory effects in this region, and it's true bilaterally, it's true in the right IFG as well, though I'm not showing it, we see that compared to items that go on to be missed in the memory task, those that are correctly recognized but where the source is incorrect show somewhat higher activity. And the highest activity goes for those items that are both recognized and where people can remember the encoding task. It's a graded effect. And there's very little evidence, a little bit of evidence, but relatively little evidence for an age effect on the magnitude. When we look at the relationship between these subsequent memory effects and memory performance, we see again only an age-dependent effect. The relationship between subsequent memory effects and subsequent memory effects and performance is unique to the older participants. And we find it as strongly for item memory as we do for source memory. So,
When you do the contrast, how much signal do you have in the contrast?
I can't hear you, sorry.
How much signal do you have in the contrast? So, like, say you have 10 items that are, and what do you subtract from that? And how many items are there?
Depends on the subject and it depends on the contrast.
Do you ever think about equating the amount of
We've done that, makes no difference at all. So we've done experiments where we randomly eliminate the excess items so that we're comparing the same mean signal from the same number of items. We get the same results. Okay, so again, age-dependent effect, very robust, same brain region, very, very different way of operationalizing episodic memory. So we're fairly confident in this case that the IFG is a robust predictor of episodic memory performance, but in older people only. In this particular study, the hippocampal effects are there, but not to my satisfaction, so I'm not showing them. There were, I won't, yeah. That's got you all thinking. Okay, moving on. At retrieval, we're now looking at the retrieval data from the associative memory task. And what we can see here now are regions of the brain that are more active when people succeed in recollecting a word pair than when they fail to recollect it. And you can see widespread, though regionally circumscribed, activations here all over the brain. These effects are, again, age independent to a first approximation. The small effects of age that we do see in these sorts of tasks go away as soon as we control for memory performance. So we don't see anything that's inherently different across age groups. This pattern of activation has been recognized for a long, long time in cognitive aging of retrieval tasks. And we refer to, everyone has to have their own network. And we felt left out because everyone else had a network. So we call this the core recollection network. It overlaps, but is not synonymous with the default mode network. And it's the same network that Dan Schachter refers to as the core network. But it's essentially a very, very characteristic pattern of activation at successful retrieval. Medial prefrontal cortex, posterior cingulate, retrosplenial, hippocampus and associated regions, and lateral parietal cortex, middle temporal cortex. We see this over and over and over again. In this experiment, I've just mentioned that we see very, very little in the way of differences in retrieval effects across age. I don't expect you to memorize or even pay very much attention to this, but the bottom line is wherever you look, age effects are either non-existent or they're slight and go away when you control for performance. This is another experiment from another study, independent set of subjects, in which we used a different way of operationalizing recollection, what's called the remember no task. This is where people are given items that they've seen before. They have to introspect and tell us whether they recollect something about the circumstances of their original presentation or whether the item just feels familiar to you. All the items are recognized. And you, again, can see the same network is activated in this very, very different sort of operationalization of episodic memory. And once again, there are no significant age effects in the size of these recollection effects. Notice that although the recollection effects themselves do not differ in magnitude, the main effect, the mean bold signal, does differ. It is higher in older people than younger people. That is orthogonal to the contrast between the conditions, which does not differ with age. And in our hands, this is a very, very common finding. The effects of age on the bold signal often take the form of a more positive going bold signal in older people than younger people. But once you look at a contrast within that condition, the age effects go away. We focus on contrast because we think they're theoretically much more important. And so when I say no age differences, I'm referring specifically to the contrast, not to the main effect. The same thing is shown here. This is, again, yet another study. It's actually a resurrected study. It's a good example of the silver lining of the pandemic. We went back and reanalyzed data that we collected quite a long time ago. In this particular study, we had a more sophisticated version of the remember no task. And the critical contrast that operationalizes recollection is between items that people introspectively say they recollect and items that they give a highly confident old judgment to, along with the declaration, I don't know why I'm doing this. I can't recollect anything about the prior occurrence of this item. And again, wherever you look, you see a reliable recollection effect, and this effect again is age invariant. But notice again, main effect of age in the signal, but no effect of age on the size of the recollection effect. If we look at individual differences, we consistently find that in the hippocampus, but usually not in other regions of the core recollection network, hippocampus only, there is a relationship which is age independent between the size of the hippocampal recollection effect and recollection performance. So this is the data from the associative recollection task that I described. This is the data from the remember no task. Very, very consistent results. It seems that in the hippocampus, whether you're 20 years old or whether you're 70 years old, the more your hippocampus turns on when you succeed in recollecting than when you don't, the better your memory is. And this does not seem to be something that differs across the lifespan. Finally, I mentioned that we operationalize something called post-retrieval monitoring. Post-retrieval monitoring is a theoretical construct which is essentially the ability that we have, having interrogated our memories and got some kind of answer back from our memories, to monitor that return signal for whether it is sufficiently strong to allow us to make a behavioral response or not. I can go into more detail in Q&A if people are interested, but that's essentially the idea. And the characteristic regions that come up again and again for various kinds of monitoring contrasts are the right dorsolateral prefrontal cortex and the anterior cingulate. And this shows the relationship between monitoring-related activity and memory performance in the associative recognition task, the RK task, same deal. Totally age independent. Doesn't matter whether you're 20 or whether you're 70. The more you can turn these regions on, the better your memory performance is. And we assume that's because you're having a more efficient monitoring process. And again, in our hands at least, retrieval monitoring does not differ either neurally at least, or in terms of its relationship with memory performance as a function of age. Even though we're looking at a region of the brain that, according to dogma, is highly sensitive to cognitive aging effects. So far we've been talking about cross-sectional data, which is okay, but of course longitudinal data is better. And so we so far have only done one attempt at a longitudinal study, which comes from the associative recognition data that I was describing to you. What we did was, in our older people, in the associative recognition task, we had them do the task here, and we took neuropsychological test scores from these individuals over a short time interval, and then a little while, 3 years later, and we had 55 people who came back after 3 years, and we asked the question whether we could find a relationship between recollection-related activity in the hippocampus at the time of scanning and memory performance 3 years later. And the answer is that we could. So the short, briefly speaking, this is memory performance on an offline neuropsychological test battery. And the memory performance was operationalized by the principal component that explained variance across different memory tests. And what we find is the bigger your hippocampal recollection effects, it was bilateral, but this is showing the left hippocampus. The larger these recollection effects at baseline, the less your memory declined over 3 years. So we found that very interesting, and we're very excited about that, and we want to follow it up. It suggests that these recollection effects that we get in these older people are telling us something about the integrity of the hippocampus, which is going to be predictive of how well you're doing down the line. And so we'd like to follow that up. I should say I'm showing here a partial correlation. The actual analysis is done using linear mixed effects modeling, simply because that's the fashionable way to do it, but you get exactly the same results if you use a partial correlation coefficient. But there you are. Okay, so that's the first part of my talk. I'll come back to the implications of that later. Just want to finish by talking about neural selectivity, which is the same thing as Thad described as neural distinctiveness. Thank you, Thad, for doing all the heavy lifting on explaining what that is. We have done a few studies looking at selectivity, two in particular that I'm going to focus on today. And so in one of these experiments, headed up by Josh Cohen, people saw pictures of objects and scenes in the scanner. We only scanned them at encoding. And they saw these pictures of objects and scenes, and they were required to make one of two kinds of judgments on these stimuli. They were cued just before each stimulus. And then at test, we showed people old and new items, and we asked them to judge whether the item had been presented in one or another of the encoding tasks or whether it was new. In the second experiment, people were scanned both at study and retrieval. This is the study task. People saw pictures of scenes or pictures of faces, and these were paired with words. And their task was to imagine where the object denoted by the word might have been in the scene, or where the object denoted by the word, how it might have interacted with the owner of the face. So with a raccoon, you might imagine a raccoon climbing a building, or the raccoon jumping up the leg of the unfortunate person whose face is depicted here. And people made vividness ratings. So one thing I should emphasize right at the outset here is, unlike the great majority of studies looking at the effects of age on neural selectivity, in our tasks, the task that people are doing, whilst the stimuli are being presented to them, are highly attentionally demanding. They require a lot of attentional engagement, and we're measuring online, item by item, how well people are processing those stimuli. We think that's crucial. What are the results? They're very easy to demonstrate. These are the results from the first of these two experiments. In this experiment, we looked only at one scene selective area, the PPA, and one object selective area, the LOC. And you can see quite clearly, if we look in the LOC, these are individual participants, younger and older. These are the means. We're looking here at a particular measure of neural selectivity that we call de-differentiation index. It makes absolutely no difference if we use a multivoxel metric of the kind that Thad described. In our hands, these give the same results. You can see no age differences at all in selectivity for objects, and a very large age difference for selectivity in scenes. This is, and moreover, when we look at the relationship between selectivity metrics and memory performance, I'm only showing one of the correlations we got in this study, but it's characteristic of the others. Neural selectivity, again, regardless of age, is predictive of memory performance for the items. It's equally predictive, by the way, for scenes as it is for objects. And the critical point is the only region that showed this relationship was the PPA. So scene selectivity in the PPA predicts memory performance. And in fact, in this experiment, it also predicted fluid intelligence on an offline neuropsychological test battery, which is a direct replication of Denise and Thad's work. So very interesting results. Here is the results from the second study. Again, we have now 4 different ROIs. The PPA, we also had retrosplenial complex, which is another scene selective region. Then we have the fusiform face area and the occipital face area. These are 2 face selective regions of ventral temporal cortex. Again, in both of the face selective areas, no evidence at all of age-related differences in selectivity. And in both scene selective regions, a very large effect of age. Much more evidence for differentiation in younger people than older people in both of the scene selective regions, suggesting there's something interesting about scenes. Again, in the PPA only, but not in the retrosplenial complex and not in the face selective areas, selectivity predicted memory performance. So there is no question there's something interesting about this selectivity metric, which is telling us something interesting about how people are processing these items. Again, it didn't matter whether we looked at memory performance for the faces or the scenes. The relationship is the same. So it's not scene selective. It's telling us something probably about a trait of some sort, which is what Thad was alluding to. PPA only again. And once again, age invariant. Doesn't matter if you're 20 or you're 70. The more differentiated if you want to put it this way, we don't think this is the right way to put it, but the more differentiated your scene representation is in the PPA, the better your memory is. So that's the encoding story. Moving on to retrieval, as I'm sure pretty well everyone in this room is already aware, there is a very standard model of the relationship between encoding and retrieval, which guides many experiments and has done for decades. And this is the idea that there is a phenomenon called a phenomenon called retrieval-related reinstatement, which is the mediator of how we come to be able to generate a representation of a prior event. And the basic idea is at the time that we're experiencing an event, activity is set up in the cerebral cortex, and this activity is captured in the hippocampus. Afterwards, the hippocampus has a memory representation. That memory representation is not a representation of the event. It is a representation of the activity that was elicited by that event. So it's a second-order representation. When a retrieval cue comes along, that it overlaps sufficiently with this stored memory representation, it gives rise to hippocampal pattern completion, and the consequence of hippocampal pattern completion is the reinstatement in the brain of the pattern of activity that was set up when you were experiencing the item in real time. So the idea is that according to this idea, episodic retrieval carries its content because you are reinstating some of the activity that actually existed while you were originally experiencing that content. It is not a perfect mirror, by the way. This is a cartoon. Things are much messier than this. And of course, I must emphasize that we know very well that retrieval is a constructive, not a reduplicative process. So this is ignoring all of the biases that come from memory reconstruction, the filling in of gaps, etc. But it's the basic idea. So it makes the obvious prediction that you should see if the only thing that's driving the precision and the fidelity of a retrieved memory is this activity at the time that you experience the memory, there should be a pretty clear relationship between the two. And in the case of the effects of age, it leads to the prediction that we should be seeing age differences in reinstatement in the same brain regions and for the same sorts of events that give rise to age differences in selectivity at the time of retrieval. We've looked at this in a couple of different ways. In an early experiment, well, early relatively, 2016, I think we published this, we had people lying in the scanner. They studied pictures and they studied words. And then they did a memory test in the scanner where they did a remember no new task on a bunch of words. Some of these words referred to the original studied pictures. Some of them referred to studied words. People were not asked to make that discrimination. They simply said, I recollect this, or I think it's old, but I don't recollect anything. And we used a multivariate classifier at the time of study to tell the difference between pictures and words, and then we applied that classifier to the test data to ask whether or not we could decode from participants' brains whether they were recollecting a word or recollecting a picture. We've done this in several ways. I'm just going to focus on this feature set here, which was selected by a univariate analysis of reinstatement effects. It doesn't matter. We've done it with ROI-based analysis. You get exactly the same results. The results are very simple to describe. When we train the classifier on subjects, within a subject, and then test it on held-out trials from that same subject, we see strong evidence of retrieval-related reinstatement. The classifier here is very accurately decoding whether you're recollecting a picture trial or a word trial, but there's no difference between older and younger people at all. Interestingly, we did this in a different way. We actually trained the classifier on all but 2 of our participants and then tested the classifier on the 2 held-out participants. This is a much stronger test of reinstatement. And remarkably, the reason we did this was we thought that we might find that older people had more stereotyped patterns of retrieval than younger people, and therefore training a classifier on older people would allow better performance on held-out older people than training on the younger subjects. The bottom line is it made no difference at all. You can train a classifier on 46 healthy older people, and then decode the memories of the two held out people very successfully. And there are no age differences, and it didn't matter whether we trained the classifier on older people's data or trained it on younger people's data. It made no difference. So this is, I think, strong evidence that at least under some circumstances, age exercises a relatively modest effect, if any, on retrieval-related reinstatement. At the time of retrieval in the second experiment, we again were able to look at reinstatement. So here, if you recall, I said that we had a study phase where people saw words paired with scenes and objects. At retrieval, people only saw the words. And they were asked to recognize the word, and if they recognized it, then to tell us, was it paired with a scene or was it paired with a face? And we were able to take two ROIs in which we got robust univariate reinstatement effects. And when we used multivoxel pattern analysis to look at the specificity or the selectivity of those reinstatement effects, we recapitulated the encoding data. No difference at all in reinstatement effects for faces. These effects are reliable, but they don't differ as a function of age. A dramatic difference in the case of scenes. People are, younger people are reinstating from their memories scene information with more selectivity than older people are. Strikingly, if we look at the selectivity with which people were encoding these items, this is for faces and this is for scenes, and we look at the selectivity with which they were reinstating those items, there is an extremely strong positive correlation. The correlation is about 0.7. And again, it turns out it doesn't matter about age, and it doesn't matter whether you look at faces or scenes. The more selectively that a face or a scene was represented when you were encoding it, the more selectively do you reinstate it when you retrieve it. And in fact, this effect is so strong that if we covary for neural selectivity at encoding, this age difference just completely goes away. So the age difference in reinstatement in this experiment is entirely determined by the fact that there are age differences at the time of encoding. Very quickly and finally, reinstatement is more than just reinstatement. Someone mentioned this morning, I can't remember who it was, I think it was Angela actually this afternoon, about something called the anterior shift. The idea that as we represent information more conceptually or in a more gist-based way, brain activity seems to move anteriorly relative to when we're representing it more literally. We looked at this, the we meaning Sabina. We means someone else, and us means not me at all. That's the general rule. But we looked at this in the retrieval data I've just described to you. I won't go into the methodology, but let me just say that what we did is we analyzed the encoding and the retrieval-related data in such a way that we could, on a participant-specific basis, identify reliably the peak region where we saw encoding-related selectivity effects and the peak region where we saw reinstatement effects. And we did this not just for the parahippocampal place area, but we did it for 4 different ROIs. I won't go into this. Suffice it to say that we find highly reliable anterior shift effects, not just in the PPA, but in all of our ROAs, the occipital place area, the retrosplenial complex, the midline face area, and the PPA. These effects are shown here. This is the mean effect across all ROIs of the anterior shift. And strikingly, this anterior shift is robustly larger in older individuals than in younger individuals, just as would be expected by the standard idea that older people retrieve information with more, in a more differentiated way than younger people do. Moreover, the bigger the shift, again, it didn't matter whether you were an old subject or a young subject, the bigger the shift, the worse your memory was, which again is understandable. So very, very quickly, to summarize, even with relatively modest sample sizes, task-based fMRI can give rise to reproducible and, I argue, theoretically informative findings. Age-invariant relationships between neural metrics and cognitive performance are very common. Age-dependent effects can also be identified. And I think it's extremely, in our hand, we think there's a very parsimonious explanation for this. Age-dependent effects, we think, can be understood in terms of a performance resource function. This is not, to put it mildly, a million miles away from Patty's crunch model. But the basic idea is we can take a brain region and think of it as having a performance resource function. If you're in the flat region of the performance resource function, you can lose brain power and you'll have no effect on the efficacy of whatever it is that that brain region is doing. We think that when we see these age-dependent effects, we're looking at regions where the performance resource function is such that younger people, their variation of neural resource stays within the flat region of the performance resource function. But in older people, we're dropping down into the what's called resource-limited region. In the case of age-invariant effects, the performance resource function simply doesn't flatten out. So you get a hit to performance whether you're a younger person or an older person if you have less than optimal neural resources available. And that's the basic explanation. Finally, neural correlates of successful encoding and retrieval we find to be remarkably stable over the adult lifespan. That needs a caveat, adult lifespan when you highly select your older individuals. Attentionally demanding tasks are employed. Age-related reductions in selectivity are difficult, at least for us, to identify other than for scenes. And we think this is an important constraint on generic models of age-related dedifferentiation, like cortex-wide changes in GABA or cortex-wide changes in dopamine availability or cortex-wide changes in signal-to-noise. How do you reconcile those sorts of models with a regionally selective effect of dedifferentiation? And as I've just said, we think, basically someone said this earlier, garbage in, garbage out. If you want to have a precise memory, it's a very good idea to encode that information precisely in the first place. We can't find any evidence that age differences in reinstatement are mediated by anything other than age differences in how you represented the information when you were experiencing it. I won't go through those three important caveats. The data are nearly all cross-sectional. There are, to my knowledge, no longitudinal studies of neural selectivity. Thad may correct me, but we know of a couple of resting state studies, but we don't know of any task-related longitudinal study of selectivity.
We're doing one now.
Apart from one that's being done now. Well, we're doing one too, so there. Yeah. Secondly, as I said, our samples are highly selected, and moreover, we do not extend into the upper age range. It's quite clear that there's a pressing need to take this sort of work into people in their 80s and their early 90s. The people we're studying and calling older are not older by contemporary standards. We've got to change that. And finally, there are various ways of doing this, but some of the work I showed you today, although we have ways now of fixing this, are subject to the criticism that we are confounding age differences in cerebrovascular factors with age differences in neural activity. We think we fixed that going forward. I'm happy to talk about it, but that is a caveat right now. That is all I have to say, other than thank you for your attention. Thank you, Denise. And here's to the next 10 years of CVL. Thank you.
Okay, I think we probably have time for one or two questions.
That was a truly spectacular talk, Vic, and very, very beautiful thing. One thing that I want to suggest, or that I think about, is I'm remembering this, we did a whole lot of pilot work at the University of Illinois on the basic parameters of the effect. And all of our effects that we find occur within the perceptual system in the ventral visual stream. And as soon as we had people do any processing of the image, because we were not happy that we couldn't guarantee that they had processed the stimuli, that they just looked at it. As soon as we did any specific processing, we lost the de-differentiation
Right.
effect.
We wish we'd known that. We would have been able to say so in our papers.
Yeah.
We think the explanation for de-differentiation in passive tasks has got nothing to do with de-differentiation as it's understood. We think it's because we know older people have difficulty sustaining attention when they are not being engaged by a task. And we know also, there's a lot of good work on this, that neuroselectivity is very sensitive to selective attention.
Yeah.
Even in young people, if you direct people's attention away from a face, fusiform face activity plummets. So we think that these early results of finding de-differentiation for faces and objects, as well as for scenes and houses, is probably a consequence of using tasks that confound attention with age.
Well,
So the fact is that even when you don't confound attention with age, you see these robust differences in the PPA. They're always there,
yeah.
and they're the ones that really excite us right
One
now.
particular argument maybe against that is that we found cultural effects. We find that older adults and younger adults from Western cultures show stronger effects for faces, whereas East Asians don't, because the information is
Sure.
not as contextual. So that sort of, I don't know exactly what to say, but I think that the notion that we only access the perceptual system is very relevant to disentangling this whole
Sure.
situation. But that was a truly brilliant talk. Thank you very much.
I'm sure Thad is going to weigh in. There's a question at the back there.
Yeah, a great talk. I don't totally buy that. I mean, we have done, think of our motor cortex stuff where people are doing a task all the time. We still see age-related difference. So I'm not, but that's not my question.
I will just respond to what wasn't your question.
Okay.
We have nothing to say about auditory or motor systems. That remains, we have never done an experiment in either of those modalities. We're hoping very soon to do an auditory experiment, actually, for lots of reasons, not least of which is, of course, the auditory pathway, the so-called Watt pathway, has a very similar posterior to anterior gradient of abstraction that the visual pathway does. But all bets are off there. Nothing to be said about that as far as we're concerned.
Okay, but actually what I wanted to ask you about was your take on the age invariant findings. So, and how you interpret that. So I could imagine an age invariant variable as you define it, nevertheless being crucial to explain age-related behavioral decline. So, for example, let's say that, let's take neural selectivity or whatever, which we see age-related differences in, and maybe longitudinally we will find age-related changes in. Even if that effect explains variance in the young and the old, it still seems like it could be crucially important if it declines with age and explains.
Well, this is very fair. There's a paper which I am responsible for not writing right now, which is called the spare variance paper in the lab. And the point is, it's based on the selectivity data. So the point being, when you control for performance, you are still left with large residual differences in selectivity and age differences in selectivity. But those age differences that are not explaining any performance differences. They're just sitting there. That's why we call it spare variance. I think there's many. I'm not arguing with you, because every other day we wake up thinking the same thing. So, but the most parsimonious prediction from your account is that means that when you take account of behavioral performance, you should lose the age differences in selectivity. Now, in the hippocampus, we do. there are often small differences in the magnitude of hippocampal recollection effects. And hippocampal recollection effects correlate with performance. When we control for performance, those age differences go away. There's no spare variance. In this case, though, there is spare variance. And that doesn't mean it's not functionally important. It could just mean we don't know what psychological tests to give people. But as it stands, the simple prediction doesn't hold up for selectivity.
So out of respect to the poster presenters and of all the carnivores, we should probably give the formal proceedings to the event and give one last round of applause. Do you have any announcements?
I have some announcements. Hold your applause, please.
Just in case, you might not appreciate your announcements.
There's just another couple of slides I want to show, Gagan, just a minute. So first off, yes, the poster presenters, the boards are upstairs, they're numbered. Please go and apply your posters to said boards, and we'll be up there for a good poster session. Second, the barbecue, which of course is what everyone's here for, starts at 6 o'clock, and it's on the 8th floor of the building, in the top floor of the Center for Vital Longevity. And we hope to see you all at both of those events.
The poster session's on the main floor.
I'm sorry, I should have said that. The poster session, yes, is one floor up in the main atrium. Well, you can't really miss it because there's like 36 posters there. So if you see that, you're in the right place. Okay. All right, thank you so much.